So for the past couple of years, I've been thinking about pollination. And pollination is really very diverse. It's very exciting. It, as we learned in the morning today, it's supposed to be one of the drivers for the massive diversity in both plant and insect forms. And over the course of these millions of years of co-evolution, what has happened is that insects and plants have both evolved extreme specializations in terms of the kind of odor molecules or volatiles the plants produce, or the kind of spectral color that flowers have to attract these insects. And insects in turn have evolved sensory specializations to take advantage of these, these signals. The ability of insects to find and feed from these flowers using these sensory systems really shapes pollination, and that has attracted a lot of attention from researchers across generations, right? So we know a lot about how insects use odor to track odor blends over large distances and find their host plants. We also know a little bit about, and we continue to learn more about how, when the insect comes closer to a flower, how it uses innate color preferences, maybe even humidity, odor, CO2, all of those to choose a particular flower. In fact, even when they've chosen a flower, they still have the task of finding the small, tiny nectary hole in which they will get the final reward. And even at this stage, the flower provides several sensory cues like color patterns on the flower petal, or mechanical texture grooves on the surface of the petal, or even just the curvature of the corolla, all of which improve the success with which the insects can find the nectary. But if you think about what the task entails, is actually quite complex. And this is especially so for insects that hover while feeding, which is the butterflies and moths, the lepidopterans primarily. So what they do is, when they approach flowers, they have these really long mouth parts called the proboscis, which they keep coiled up. They uncoil it, and as they approach the flower, they are typically uncoiling it. And this is a video of a Madagascar hawk moth. Its proboscis is about 5 times its own body size. And this video will sort of let you know a little bit about how complex this behavior is and how much control it might need to actually get into the nectary, right? So it has a really long proboscis, which is quite flexible, and somehow it has to get it inside into a very tiny hole. And it's doing all of this while it's hovering, typically when the flowers are swaying in the wind. Not just that, if you think about moths, which have longer proboscis, they're doing this at night times when visual stimulus is limiting, right? So they're really up for a challenge. The other thing that was really exciting about these insects were that they have a really unique proboscis itself, the mouth part. So this is a moth head, and if we take a cross-section, virtual cross-section of its proboscis in its coiled state, what you'll see is that it's actually made up of 2 half tubes. The outer tubes that are drawn here in gray or blue are the ones that are filled with hydraulic fluid to uncoil it. The inner tube is what serves as the drinking straw. Now this outer tube is lined with muscles. It's lined with muscles at the proboscis base as well as along its entire length. That's the red and the green muscles. And there are a whole bunch of other set of muscles in pink, orange, and blue that are involved in pressurizing and moving the fluid into the proboscis. And this is what is controlling the motion of the proboscis. In addition, the proboscis itself has a lot of chemical as well as tactile sensors, potential tactile sensors, and here I've just shown you sense of a few of them. Then the top one are sensors that are potentially at the base, potential sensors that are at the base of the proboscis, just at the joint between the head and the proboscis. And then they're all along the length. And there are a variety of these sensors, right? So today what I'll briefly tell you about is how these insects might be using the proboscis to actually target nectary, especially how would they be using the tactile cues. We'll have a little discussion about how they might integrate this with other visual cues and what this means in the context of pollination in general. And so this work started out at the University of Washington as an HFSP grant with Tom and Bing Brunton. And there was a very talented undergrad that we recruited, Mahad Ahmed, who contributed significantly to these projects. And so here we started out asking very simple questions, which is that we know that all these features on the flower really help moths reach the nectary. But are these insects actually using these cues? Are they actually acquiring these cues and somehow using it to get better at finding the nectary? Is there any evidence of them actively searching, or there's some evidence of active sensing, or are they just randomly poking on the flower surface until they find the nectary? To do this, the first thing we did was we set up a behavioral assay. So we got the behavior into the lab, and this involved using artificial flowers. So we had with 3D printed flowers, which means now we can provide whatever stimulus we want. And at the base of that 3D printed flower, we provide an artificial nectary, a very small quantity of sugar solution, which we combine with in-house microsensors. So this is a small motion sensor at the base, which detects when the proboscis comes in. We connect this with an automated pump. So every time the moth comes, feeds, goes away, you fill the nectary back in. And what that allows us to do is then the moth can repeatedly visit this flower and again, and we can track what it's doing with its proboscis, as well as how that changes with learning, or if there is learning. And the other advantage is because it's artificial flowers, we can provide gradients of sensory cues. We chose to first look at curvatures, and so we provided flowers which are very easy to exploit, funnel-shaped flowers, all the way to a flat flower which just has a hole in the center, which is very hard to exploit. And so, and what we do is we have a camera on the top which is looking at all of this interaction. So what we did first was we quantified, we tracked the tip of the proboscis, which is really tiny, and we plotted what the trajectory of this proboscis tip looks relative to the flower radius. So what I have over here is time on the x-axis, and on the y-axis, 0 is the center of the flower, 1 is the edge of the flower. And what we see is that as the moth explores the flower, it's definitely not randomly poking the flower. In fact, it's doing very systematic searches. It's going from the edge of the flower towards the center, back to the edge and center. Right? So it's doing this really systematic search until it finally eventually finds the nectary. If we look at what it does across different kinds of flowers, what we see is that even for the flowers that are hard to exploit, the flat flower, it uses similar strategies, except it's doing it more extensively and longer because it's much harder to find. We looked at what this means in terms of do they change how they find the flower or the nectary with repeated visits. So is there any evidence of learning? So what we plotted was on the y-axis again is exploration time, which is time taken to find the nectary from when it started exploring, and on the x-axis is visit number, and if it goes down, that typically means that it's learning. So if you compare the early visits, which are in orange, to what you see in green, what you see there is that the exploration times have reduced, suggesting that the animals have learned how to explore these flowers. And if we look at across flower shapes, what you see is across different flowers, the learning varies. So harder flower is difficult to exploit during the early visits, but in the later visits, even if you have a slight sensory gradient, they will learn really quickly. And so now we're very excited to understand whether this varies or how does this extend if your texture of your tactile cue varies. So if you have textures versus edges, if the nectary is not in center, how do these exploration patterns vary? Are they context dependent? And how those might be controlled? And so very briefly, I think I have only 13 seconds left, is that correct? Okay, so I'm just going to talk about, we also tried to see what this might mean if you So we talked about different kinds of texture or different kinds of tactile cues, but remember they're actually using a whole bunch of different kinds of cues. So we want to look at the role of vision here, and we took advantage of the fact that these moths are active during a large range of light levels. So the data that I showed you were at 0.1 lux, which is cloudy night. We did the same experiments at 50 lux, assuming that more light means more visual stimulus, improved spatiotemporal resolution, and hence maybe better performance. And what we actually saw was quite the opposite. So if you look across the two light levels, the number of visits remained the same, but the fraction of successful visits actually lower for the higher light levels, despite better visual acuity. I keep pointing in the wrong direction. And in fact, at the higher light levels, there is no learning at all, unlike what we saw earlier. Thank you. So what we've tried to explore this, and we've seen that the body kinematics is actually not quite changing. The way they're flying is similar across the two regimes, and we think that it's the exploration of how they're moving the proboscis that is changing. And so we're going to manipulate different aspects of visual cues, maybe the contrast, to see how visual and mechanosensory cues combine. I'm going to skip the last part completely, which was talking a little bit about the work we've started doing at Shiv Naimal University. Sorry, Ananya. So ask me about it. But I'll just move on to say we are recruiting. So if you're interested in doing this kind of work, please do come and talk to me. There's another graduate student from a neighboring cancer lab, Shaisa Heather, who's at poster number 16, so go find her if you're interested in knowing life at SMU. She'll also tell you about her ideas of using evolutionary theory to treat cancer. And with that, I would like to thank you, and especially the organizers, for giving me a chance to talk over here.
Thank you so much, Tanvi. That was a very interesting talk. Do we have questions? Sumana.
Would there be a relationship between the reward? So, I mean, better reward leads to learning that is faster, things like this, or not really?
We haven't explored that in lepidopterans, and in fact, I know that has been explored a little in bees, but I don't know of anybody looking at either butterflies or moths. The thing is that most of these flowers provide very small amounts of reward. You know, typically the nectar amounts are very tiny. But I don't know if
quantity may be lower, but the quality can be different.
Absolutely. And in fact, it will vary at different times of the day as well. So we haven't looked at that in terms of their motivation of learning. That's interesting. Yeah.
Oh.
Yeah, I was wondering what is the quantity of syrup in your artificial nectar?
So we put about it's a 20% sucrose solution that we give them, and the quantity is about 50 microliters.
Is that comparable to what you find in the
Yeah,
lab?
so we've tried to mimic.
Microliters.
So we've looked like the people have actually studied that in the field, and we've tried to keep that comparable.
Hi, nice talk.
Thank you.
Any flower is known to have some kind of a tactile guide kind of a thing?
Yeah. So almost all flowers that are pollinated by bees or lepidopterans, so hymenopterans and lepidopteran flowers will have visual and tactile guides. Visual guides, by visual guides I mean they will be either colored stripes on the flower that leads to the nectary, or they will have a bull's eye that marks the nectary position. They will have textures on the flower, so differentially textured texture that leads you into the nectary. And if you mismatch that, if you artificially sort of misplace the texture with respect to where the nectary is, the animals do much worse, which is how we know that they might be using it.